guys, so now we're talking about gel electrophoresis, or as you may know it, DNA fingerprinting. So, first I need you guys to remember that our DNA varies, but only slightly. So 99.9% of your DNA is the same as the person sitting next to you right now. So there's a lot of differences if we look at it very closely, but the majority of our DNA is the same. So what we have to focus on are sometimes these parts called introns or non-coding regions of our DNA where we have these differences. So what happens first in order to do a DNA fingerprint or to do gel electrophoresis is scientists will cut the DNA with a special enzyme called a restriction enzyme. Now I'm not going to let you forget, enzymes are molecular scissors, so that's why they can cut the DNA. So it'll recognize the DNA at a special sequence. So for example, it'll recognize the DNA right here at the CGGCAT, and then it'll cut the DNA right there in kind of a zigzag fashion. And then we'll end up with two fragments of DNA of different sizes, or more, or however many times it recognizes this sequence in the DNA. All right, so then what we can do is we can either get different samples of DNA with the same restriction enzyme and compare the results, or we can use different restriction enzymes to cut the DNA and see what happens. And sometimes we'll cut the same DNA with multiple restriction enzymes just in order to get a variety of fragment lengths. So for example, if I were to cut my DNA, ooh, this is a terrible marker. If I have my DNA strand here, if I were to cut it with my restriction enzymes and it made 3 cuts because it recognized the specific site 3 times, so 1, 2, 3, how many fragments would I have? Well, this cut here, and this cut here, that gives me 1, 2, 3, 4 fragments. Now they wouldn't probably all be that even. Sometimes if we cut a piece of DNA, the restriction enzyme will recognize a site here and maybe a site at the very end. So we'll have a tiny little fragment at the end and then one here. And you notice our fragments are not the same size, and this is where our gel electrophoresis is going to be very helpful. So we can cut our DNA with a variety of restriction enzymes, such as BAMH1, Hindi3, EcoR1. There's a ton of them out there. And then we're going to use a special material called an agarose gel, which is sort of like a matrix. So the DNA can go through these tiny little pores in the matrix if we run a current down the gel. Now, it's important to remember that DNA is negatively charged, so it will be attracted to the positive end of the current and it'll slowly travel down the gel. So we'll load our DNA at the top. Sometimes we'll load, we call a marker here in the first one, or our evidence. We'll load DNA here, we'll load DNA here. These are called our wells. We'll do this very carefully. And then as the current starts to run, the DNA will travel down the gel, and based on the length of our specific fragment, it will go further or shorter. So think of a large fragment as like a really big fat guy who can't run a long race, and a tiny little marker is like a tiny little skinny person who's going to run really fast and go all the way down and travel further through the gel. It's just easier for small fragments to travel further. So maybe we'll have a fragment down here, and a fragment here, and a fragment here.
But this DNA will have a fragment here, here, and here, and this DNA here, here, and here, and here, here, and here. Oops, too many.
And then we're going to see which one matches closely to our marker or our evidence or whatever we chose to put in that first well. And if we look at, we can line these DNA fragments up. So there's a match, and here's a match, and here's a match, here's a match. And it looks like these two were the best matching fragments. So we can probably conclude that either these two have some relatedness, maybe they belong to the same person, maybe it's evidence that matches at a crime scene, but there's lots of applications for gel electrophoresis and DNA fingerprinting. For example, we can use it to find missing or identify missing persons. Of course, we can use it in crime scene investigation, but also in paternity testing or looking at different organisms and seeing if they have any evolutionary relatedness. So there's a lot of great applications. Now, I want to talk really quickly about the ethical considerations for doing DNA fingerprinting on a regular basis, and it's used very frequently now in a lot of different crime scenes, but you need to remember, in order for there to be DNA fingerprinting, we need to have a crime scene with nucleated cells, meaning DNA is still within the nucleus of the cells, or else you don't get any DNA. All right, so first question I want you to think about, on what basis should people be required to submit a DNA sample in crime scene investigations? What are some potential sources of error in such an investigation? And do you think DNA fingerprinting alone provides substantial enough evidence for a positive identification of a criminal. All right, just some questions for you guys to think about it. We'll talk about them in class. Thanks.